Dear friends, once again hearty and warm welcome. Friends, in this lecture today we are going to learn new topic, that is DNA replication. As all of we are known that DNA is a genetic material and it regulates and controls all the different cellular activities including synthesis of different biomolecules. I said that DNA as a genetic material responsible to regulate and control all the different cellular activities, as well as it regulates, it directs, it controls the synthesis of different biomolecules, especially the synthesis of proteins, synthesis of RNA, as well as it regulates the synthesis of DNA itself, and on the basis of which there are two major functions of DNA. Accordingly, there are two major functions of DNA, such as autocatalytic functions and heterocatalytic functions. Let me explain you what does it mean. First of all, heterocatalytic function. Heterocatalytic functions of DNA. Heterocatalytic functions of DNA means the DNA when regulates, controls the synthesis of other biomolecules except DNA, such as the synthesis of RNA, synthesis of proteins, then it is known as heterocatalytic functions. Very simple. Heterocatalytic functions means when DNA regulates or controls the biosynthesis of proteins and RNA, then it is known as heterocatalytic functions. And when the DNA regulates, controls the synthesis of DNA itself, when it controls, regulates the synthesis of DNA itself, then it is known as autocatalytic functions. Autocatalytic functions. You know, here hetero, it stands for other or different, and auto stands for self. So whenever the DNA, it regulates the synthesis of DNA itself, known as autocatalytic functions, and when it regulates the synthesis of other biomolecules except DNA, that is RNA or proteins, then it is known as the heterocatalytic functions. Now, the autocatalytic function involve the synthesis of DNA itself, and actually this process is nothing but the replication. Replication is a process in which DNA produce exact copy of itself. Replication is a process in which the molecules of DNA duplicates. The content of DNA in the cell get doubled, and this process in which the parent DNA molecule produces daughter DNA molecules which are exact copies of itself is simply known as DNA replication, or it may be known as DNA duplication, because during this process the content of DNA, amount of DNA in the nucleus get doubled, hence it may be known as the duplication. So once again, let us talk about the definition of replication. I said just now that replication, it is a process in which parent DNA produces daughter DNA molecules which are exact copies of the parent DNA is known as DNA replication. Now, the process of DNA replication in eukaryotes first of all discovered by those great scientists, that is Watson and Crick, in year 1953. Now let me tell you that the replication, it takes place in the nucleus because the DNA is present in chromosome and chromosomes are present in the nucleus of cell, and therefore in eukaryotic cells the replication takes place in the nucleus, and the process of replication, the process of duplication of DNA takes place during the S phase of interphase of the cell cycle. Listen, the replication, it takes place during the S phase, synthetic phase of interphase of cell cycle. We all have studied about the cell cycle in 11, isn't it? We know that the cell cycle consists of two main phases such as M phase and interphase, isn't it? Here I am going to show diagrammatically. Suppose here is the M phase and here is the interphase. We have seen that the interphase is further divided into G1 phase, S phase and G2 phase. Okay? During M phase, it occurs the division of cell leading to the formation of two daughter cells, and in interphase that newly formed cells are again prepared to undergo the next cell division. Now here in this S phase, S stands for synthetic or synthesis phase. In this phase actually the DNA replication takes place. It means the content of DNA in that cell is doubled because it is necessary for the equal distribution of DNA during the process of cell division. याचा अर्थ की replication ही प्रक्रिया न्यूक्लियस मध्ये घडून येते आणि प्रत्यक्ष replication हे सेल सायकल मध्ये S फेज मध्ये घडून येत असतं. कारण ज्या वेळेला आपण सेल सायकल बघतो त्यावेळेला सेल डिव्हिजन झाल्यानंतर ज्या डॉटर सेल्स तयार होतात त्या पुन्हा इंटरफेज मध्ये प्रवेश करतात. इंटरफेज मध्ये पुन्हा त्या सेल्स तयार होतात, प्रिपेअर होतात फॉर नेक्स्ट सेल डिव्हिजन, आणि ह्या इंटरफेज मध्ये एस फेज मध्ये सेल असताना डीएनए चं रेप्लिकेशन होतं, डुप्लिकेशन होतं. याचा अर्थ डीएनए चं कंटेंट हे डबल केलं जातं. कारण ज्या वेळेला पुन्हा त्या सेलचं सेल डिव्हिजन होईल त्यावेळेला डीएनए चं डॉटर सेल्स मध्ये इक्वल डिस्ट्रीब्युशन होणं हे गरजेचं असतं. अँड दॅट्स व्हाय आय सेड द रेप्लिकेशन इट टेक्स प्लेस इन द एस फेज ऑफ इंटरफेज ऑफ सेल सायकल. नाउ we people have to study about the mechanism of replication, but before to that one must know the basic requirements for the process of replication. So let me tell you which are the basic requirements for the process of replication. Basic requirements for the process of replication. First, DNA template. DNA template. DNA template means it is the strand of DNA on which new DNA strand is to be synthesized. DNA template means the strand of DNA on which new DNA strand is to be synthesized. याचा अर्थ ज्या DNA strand वर नवीन DNA strand हा कन्स्ट्रक्ट केला जाणार आहे, सिंथेसाइज केला जाणार आहे, तयार होणार आहे, अशा ओल्ड स्ट्रँडला टेम्पलेट स्ट्रँड असं म्हणतात किंवा याला मोल्ड स्ट्रँड असं पण म्हणतात. मोल्ड मीन्स इट सपोज टू प्रोव्हाइड साइट फॉर द सिंथेसिस ऑफ न्यू स्ट्रँड. सेकंड, DNA polymerase enzyme. This enzyme is necessary for the synthesis of new strand on the old strand. Listen carefully. DNA template means the old strand on which the new strand is to be synthesized, and for the synthesis of new strand, whatever important enzyme is necessary known as DNA polymerase enzyme. RNA primer, it is a short segment of RNA, or it is a short segment of ribonucleotides. Short segment of ribonucleotides. It is necessary to initiate the process of DNA replication. RNA primer हा RNA मॉलिक्यूलचा छोटासा सेगमेंट आहे आणि हा प्रत्यक्ष रेप्लिकेशनची प्रोसेस सुरू होण्यासाठी त्या ठिकाणी आवश्यक असतो. And for the synthesis of this RNA primer, again it requires another enzyme known as RNA polymerase. RNA polymerase. Next enzyme is helicase. Helicases. This enzyme is necessary for the unwinding or uncoiling of DNA double helix. Uncoiling or unwinding of DNA double helix. We know that DNA molecule is made up of two strands which are coiled around each other. So, for the process of replication, initially the unwinding, uncoiling of that two strands is necessary, and it is brought with the help of enzyme helicases. Next, helix destabilizing protein. It may be called as single strand binding protein, SSBP. What is the role of this enzyme? I am going to explain you, but temporarily let me tell you that this enzyme or this protein, it prevents the recoiling of two strands of DNA. All the four types of deoxyribonucleotides, all of we are known that DNA is nothing but the polynucleotide molecule. DNA is made up of two long chains of deoxyribonucleotides. Four different types of deoxyribonucleotides are necessary. They act as the building block of DNA, such as deoxyribose adenine nucleotide, deoxyribose guanine nucleotide, deoxyribose cytosine nucleotide, deoxyribose thymine nucleotide. And here these deoxyribonucleotides are present in the nucleoplasm, freely floating in the nucleoplasm, so they are necessary for the synthesis of new DNA strands. The magnesium factor is also necessary as a cofactor. So these are few of the important basic requirements for the process of DNA replication. Now actually we are going to move towards the mechanism of DNA replication, okay? But let me tell you, most commonly the DNA replication is of semi-conservative method. Semi-conservative method means what? Please listen. Semi-conservative method means it is the method in which each of the daughter molecule produced, each of the daughter DNA molecule produced shows one old strand, that is one strand of parent, that is old strand or parental strand, and one new strand. Let me explain you. Suppose here is parent DNA. Just consider. Here is parent DNA, double-stranded DNA. Parent DNA. And as it undergoes replication, it produces two daughter DNA molecules which are exact copies of the parent DNA. Now in these daughter DNA molecules, one strand comes from the parent. One strand is parental strand, that is old strand, and one strand is newly synthesized strand. Means according to this diagram we can say here in these two daughter DNA molecule, suppose this daughter molecule has received this pink colored strand from the parent and yellow colored strand is newly synthesized. While here in this other daughter DNA molecule, yellow strand is suppose parental old received from the parent DNA and the pink strand is newly synthesized. Means what? In this type of replication, half of the material is conserved, half of the material is parental and half of the material is newly synthesized. सोप्या भाषेत सांगायचं झालं तर हे जे दोन डॉटर डीएनए मॉलिक्युल्स तयार झाले आहेत त्यांच्यामध्ये प्रत्येकाजवळ निम्मी 50% 50% हे वडिलोपार्जित संपत्ती आहे, पैतृक संपत्ती आहे. म्हणजेच काय, पॅरेंट कडून मिळालेला एक स्ट्रँड आणि नवीन तयार झालेला एक स्ट्रँड, आणि म्हणून अशा प्रकारच्या रेप्लिकेशनला सेमी कॉन्झर्वेटिव्ह मेथड ऑफ रेप्लिकेशन असं म्हणतात. नाउ लेट अस स्टार्ट द स्टडी ऑफ ऍक्च्युअल मेकॅनिझम ऑफ डीएनए रेप्लिकेशन. लेट अस स्टार्ट. Mechanism or steps of replication. First step, activation of nucleotides. Just now I told you DNA is made up of two anti-parallel strands of deoxyribonucleotides. DNA is made up of two anti-parallel strands of four different types of deoxyribonucleotides. Now, all these four different types of deoxyribonucleotides are present freely floating in the nucleoplasm. हे जे चार प्रकारचे deoxyribonucleotides डीएनए स्ट्रँड सिंथेसिस होण्यासाठी आवश्यक आहे ते न्यूक्लिओप्लाझम मध्ये प्रेझेंट असतात. बट ऑल दीज फोर deoxyribonucleotides are present in the form of their monophosphates, such as deoxyribose adenine monophosphate, deoxyribose guanine monophosphate, deoxyribose cytosine monophosphate, deoxyribose thymine monophosphate. And all these four different types of deoxyribonucleotides in the form of monophosphates are first of all converted into their triphosphates, and for that purpose the ATP molecules are utilized, and thus these monophosphates are converted into triphosphates, such as the deoxyriboadenine monophosphate plus ATP gives deoxyribose adenine triphosphate. Means what? Here the two phosphate groups get added into the AMP so that the AMP gets converted into ATP. Now that two phosphate groups are obtained or donated by this ATP molecule, and therefore this ATP molecule is converted into AMP, that is adenosine monophosphate. आपल्या लक्षात आलं असेल या ठिकाणी या ATP मॉलिक्यूलने दोन फॉस्फेट ग्रुप DAMP ला डोनेट केले की ज्यामुळे DAMP चं रूपांतर झालं DATP मध्ये. म्हणजे deoxyribose adenine monophosphate is converted into deoxyribose adenine triphosphate, and same thing happens here about these remaining three, that is DGMP plus ATP produces DGTP plus AMP, DCMP, that is deoxyribose cytosine monophosphate plus ATP gives DCTP, that is deoxyribose cytosine triphosphate plus AMP, deoxyribose thymine monophosphate plus ATP give rise to deoxyribose thymine triphosphate plus AMP. Now here all these four different types of deoxyribose nucleotide molecules in the form of monophosphate gets converted into their triphosphate by the utilization of ATP molecule and in presence of enzyme phosphorylase. So actually this process is the phosphorylation of these deoxyribose nucleotide, and it is nothing but the activation of nucleotide. It means now all these four different types of deoxyribose nucleotide which are in the form of triphosphates are ready to take part in the process of replication, in the process of synthesis of new DNA strand. Now come to the second step. Second step, origin or initiation point. The process of replication on DNA, it starts at specific point, at specific region, and that point on DNA molecule where it starts the process of replication is known as initiation point or point of origin or simply origin. Let me explain it. Suppose here is double-stranded DNA molecule, eukaryotic linear DNA molecule I am going to show. Now I said that the point where actual process of replication starts is known as point of origin or initiation point. In eukaryotic DNA, the process of replication may start at more than one site, because in eukaryotes the DNA is large sized. It means in eukaryotic DNA there may be more than one initiation point, but in prokaryotes, that is in circular DNA, being small sized, there is only single point of origin, only one initiation point. Now let us consider, suppose this end is the initiation point. At this end or at the point of origin, the weak hydrogen bonds between the two strands of DNA helix are cut off. Two strands, we know the two strands of DNA are joined with each other by means of weak hydrogen bonds. So here that weak hydrogen bonds between the two strands of DNA are cut off. They are broken down in presence of incision enzyme known as endonuclease. It is called incision enzyme because it makes an incision. You know, incision means cut. Incision याचा अर्थ होतो कट. म्हणजे ह्या एन्झाईमच्या मदतीने डीएनएच्या दोन स्ट्रँड्सला जोडणारे जे वीक हायड्रोजन बॉन्ड्स असतात ते इनिशिएशन पॉइंटच्या ठिकाणी कट केले जातात, ब्रेक केले जातात, and this process is actually called as incision. या प्रोसेसला incision असं म्हणतात. So here we have studied about the origin or initiation point. Now direction of replication. Just now we have studied that at the point of origin the weak hydrogen bonds between the two strands of DNA are cut off by an incision enzyme, that is endonuclease. The process of replication is either unidirectional or bidirectional. Unidirectional or bidirectional. Let me explain you. Suppose the point of origin in eukaryotic DNA is somewhere on the DNA molecule except both ends. दोन्ही टोकं वगळता कुठेतरी मध्ये जर पॉइंट ऑफ ओरिजिन असेल, मग या ठिकाणी पॉइंट ऑफ ओरिजिन असेल, या ठिकाणी असू शकतो, या ठिकाणी असू शकतो, then the process of replication it takes place in both directions away from the point of origin. Away from the point of origin. याचा अर्थ जो पॉइंट ऑफ ओरिजिन आहे, कल्पना करा की या ठिकाणी पॉइंट ऑफ ओरिजिन आहे, या ठिकाणी दोन स्ट्रँड्स मधले जे वीक हायड्रोजन बॉन्ड्स आहेत ते ब्रेक होतील आणि नंतर रेप्लिकेशनची प्रक्रिया सुरू होईल, तर रेप्लिकेशन हे दोन्ही दिशेने होतं. याचा अर्थ बाय डायरेक्शनल. That is the replication and synthesis of new strands it takes place in opposite direction going away from the point of origin. This is called bidirectional. But rarely if the point of origin is at one end, any one of the ends, समजा पॉइंट ऑफ ओरिजिन या टोकाला आहे किंवा या टोकाला आहे, अशा परिस्थितीमध्ये the replication takes place only in single direction. Suppose here is the point of origin, then replication occurs in this direction, that is unidirectional. So here we have discussed about the direction of replication. Now unwinding of DNA strands. Unwinding, that is uncoiling of DNA strands. For the process of replication, the unwinding or uncoiling of two strands of DNA is necessary. So the unwinding and the process of replication takes place simultaneously. Unwinding याचा अर्थ काय? Unwinding म्हणजेच uncoiling. We know that both strands of DNA are helically twisted, helically coiled around each other. So their separation, their uncoiling is necessary. अर्थात या ठिकाणी पॉइंट ऑफ ओरिजिन जवळ दोन स्ट्रँड्सला जोडणारे वीक हायड्रोजन बॉन्ड कट झाल्यानंतर these two strands undergo uncoiling or unwinding and it is brought by enzyme helicases. Helicases or unwinding protein, helix unwinding protein, or it may be called as a rep protein. Here REP, rep it stands for replication. So these helicases or DNA unwinding protein or rep protein, they bring about the uncoiling of both strands of DNA so that they can be separated from each other, and the recoiling of these two strands of DNA is prevented by another enzyme, because after unwinding, after uncoiling, the recoiling of both strands may be possible. जरी या एन्झाइम्सच्या मदतीने DNA चे दोन स्ट्रँड्स च अनकॉइलिंग झालं तरी सुद्धा पुन्हा त्यांचा रिकॉइलिंग होऊन जाण्याची शक्यता असते, आणि म्हणून त्यांचा रिकॉइलिंग टाळण्यासाठी, प्रिव्हेंट करण्यासाठी त्या ठिकाणी दुसरा एन्झाइम, दुसरं प्रोटीन काम करत असतं. त्याला म्हटलं जातं हेलिक्स destabilizing protein. Helix destabilizing protein or single strand binding protein. Single strand binding protein. name itself indicate this protein molecule, it binds to any one of the separated strand so that their recoiling is prevented. Along with helicases, SSBP, one more enzyme, topoisomerase, it also assists for the uncoiling and separation of two strands of DNA. Now, after the separation, after the uncoiling of two strands of DNA, the DNA strands like a fork, just like a fork. Let me show. Or Y-shaped structure, just like a fork or Y-shaped structure. अर्थात हे दोन DNA चे strands एकमेकांपासून separate झाल्यानंतर at the point of initiation it appears capital Y or fork-like structure. It is known as replication fork. Replication fork. Now, due to unwinding, two strands which get separated from each other now acts as a DNA template, acts as a mold strand on which the new strands are synthesized. आता या ठिकाणी DNA चे जे दोन स्ट्रँड्स एकमेकांपासून अलग झालेत, सेपरेट झालेत, ते सेपरेट झालेले दोन्ही स्ट्रँड्स हे टेम्पलेट स्ट्रँड म्हणून काम करतात. त्यांना मोल्ड स्ट्रँड्स असं म्हणतात. We know that the DNA has two anti-parallel strands. So suppose this yellow strand has direction 5' to 3'. Here I am going to write, suppose 5' to 3', and this pink strand has 3' to 5'. Okay. Now this both strand acts as the template or mold strand on which the new strands are synthesized. आता या दोन स्ट्रँड्स वर नवीन स्ट्रँड हे सिंथेसाइज होणारे, DNA चे नवीन स्ट्रँड तयार होणारे, because the nitrogen bases of these both template strands are fully exposed, isn't it? Nitrogen bases of both strands are fully exposed. आता या ठिकाणी या दोन्ही स्ट्रँड्सचे जे नायट्रोजन बेसेस आहेत, मग ते ॲडेनिन असेल, ग्वानिन असेल, थायमिन असेल, सायटोसिन असेल, ते एक्सपोज आहेत, so that now they can easily receive the nitrogen bases or nucleotides necessary for the synthesis of new strand. So let us talk now about the actual mechanism of synthesis of new strand on the old strand. So the next step, synthesis of new strand. First of all, let me draw here a diagram. Let us consider here is a double-stranded DNA with two anti-parallel strands, 5' to 3', another strand 3' to 5', and just now we have studied as these two strands of DNA helix get separated from each other, there is a formation of fork or capital Y-like structure. You know, this structure is known as replication fork. Replication fork. Now, let us talk about the actual mechanism of synthesis of new strands on the old strands. The synthesis of new strand occurs on the template strands or mold strands. Try to understand. Here, this 3' to 5' is one DNA template, and this another 5' to 3' is another template. Now the synthesis of new strand takes place on these two template strands or mold strands. Let me tell you the meaning of mold. You might have listened, you might have studied, you might have heard that mold means it is a hollow container in which some molten material is filled by the ironsmith, goldsmith, or even the potters, that is the ceramist, for the construction of different articles. याचा अर्थ लोहार काम करणारे लोक किंवा सुवर्णकार असणारे लोक किंवा आपण म्हणूया की मूर्तिकार, मूर्ती घडवणारे जे लोक आहेत त्यांच्याकडे काही विशिष्ट आकाराचे साचे असतात, त्यांना आपण मोल्ड म्हणतो. आणि त्याच्यामध्ये ते विविध प्रकारचं मटेरियल भरतात, पोअर करतात आणि एक्झॅक्टली त्या मोल्डच्या अपोजिट आकाराची मूर्ती किंवा कुठलीही वस्तू त्या ठिकाणी ते निर्माण करत असतात. सो हिअर आय वॉन्ट टू टेल यू these two old strands are called as template strand or mold strand. Now for the initiation of synthesis of new strand, या दोन टेम्पलेट किंवा मोल्ड स्टँड वर नवीन स्टँड सिंथेसिस होण्यासाठी इनिशियली इट रिक्वायर्स RNA primer. I have already told you that RNA primer is nothing but a very small segment of RNA or a short segment of ribonucleotides known as RNA primer. This RNA primer is synthesized on these DNA template. हा RNA primer जो आहे तो या DNA template वरच सिंथेसिस केला जातो in presence of enzyme RNA polymerase or enzyme primase. And this RNA primer get associated to the 3' end of template strand. हा जो RNA primer आहे तो या दोन्ही टेम्पलेट स्ट्रँडच्या 3' एंडला बाइंड होत असतो. लेट मी शो इन द डायग्राम. प्लीज लुक. हिअर इज सपोज टू बी द RNA primer. Consider with the help of green chalk I am going to show the RNA primer. So RNA primer get associated to the 3' end of template strand. This RNA primer attracts the activated nucleotides already present in the nucleoplasm. We all are known this process is going on in the nucleoplasm of nucleus. ही प्रक्रिया न्यूक्लियसच्या न्यूक्लियोप्लाझम मध्ये चालू आहे आणि हे शिकत असताना पहिल्या स्टेप मध्ये मी ऑलरेडी तुम्हाला सांगितलंय की all the 4 different types of deoxyribonucleotides are activated. They are converted from their monophosphate form to the triphosphate form so that all different activated deoxyribonucleotides in the form of triphosphate, that is DATP, DGTP, etc. they are attracted towards the old strand. And the selection, appropriate nucleotides are selected and they are placed and bind against the complementary nucleotides of the old strand. Listen carefully. या RNA प्रायमरचं काम आहे की न्यूक्लियोप्लाझम मध्ये असणारे जे ऍक्टिव्हेटेड डिऑक्सिरायबोन्यूक्लियोटाइड्स आहेत त्यांना या ओल्ड स्ट्रँड कडे अट्रॅक्ट करायचं. त्यापैकी योग्य प्रकारचा डिऑक्सिरायबोन्यूक्लियोटाइडला सिलेक्ट केलं जातं अँड दॅट बाइंड्स अगेन्स्ट द कॉम्प्लिमेंटरी न्यूक्लियोटाइड ऑफ द ओल्ड स्ट्रँड. आता या ठिकाणी मी ओल्ड स्ट्रँड वर समजा इथे पहिला न्यूक्लियोटाइड आहे adenine. We know that the complementary nucleotide of adenine is thymine. So the thymine nucleotide get placed against it. Okay? And the weak hydrogen bond is established between them. If there is a thymine on the old strand, then against it adenine nucleotide is placed and weak hydrogen bonds are formed. In the same manner, if there is a C, that is cytosine on the old strand, against it guanine is placed, and if there is a guanine on the old strand, the cytosine nucleotide is placed against it, and of course the hydrogen bonds are formed between the complementary nucleotides. We know that between adenine and thymine the double weak hydrogen bonds are formed, and between guanine and cytosine triple weak hydrogen bonds are formed. Now these newly arranged nucleotides which are arranged in a sequence, they are interconnected with each other by means of phosphodiester bond. हे जे नवीन न्यूक्लिओटाइड्स या ठिकाणी अरेंज झालेले आहेत ओल्ड स्ट्रँड वर, दे आर इंटरकनेक्टेड विथ इच अदर. ते एकमेकांशी जोडले जातात बाय मीन्स ऑफ फॉस्फोडायस्टर बॉन्ड. फॉस्फोडायस्टर बॉन्डच्या मदतीने ते एकमेकांशी जोडले जातात. Ultimately there is a synthesis, there is a formation of chain of deoxyribonucleotides, that is nothing but the new strand is synthesized on the old strand. For the selection, appropriate arrangement of these nucleotides require enzymes like DNA polymerase III. DNA polymerase III. Now, here we have seen about the synthesis of new strand. Not only the DNA polymerase III, but it also requires ATP or GTP as a source of energy and magnesium ion as a cofactor. In this manner, on both of the old strand, the new strands are synthesized. Not only on this strand, but on this strand also, here there is a synthesis of new strand. Suppose these are the nucleotides and get interconnected with each other. Now, out of these two template strand, हे जे दोन टेम्पलेट स्ट्रँड आहेत त्यापैकी एका टेम्पलेट स्ट्रँडला लीडिंग टेम्पलेट म्हणतात आणि दुसऱ्याला लॅगिंग टेम्पलेट म्हणतात. व्हाय? लेट मी टेल यू. लीडिंग अँड लॅगिंग स्ट्रँड, या ठिकाणी मी लीडिंग आणि लॅगिंग स्ट्रँड लिहिलंय. पहिल्यांदा मी लीडिंग टेम्पलेट आणि लॅगिंग टेम्पलेट कोणाला म्हणतात ते सांगतोय. आउट ऑफ these two template strand, या दोन टेम्पलेट स्ट्रँड पैकी the template strand which has free 3' end, which has free 3' end, या ठिकाणी डायग्राम मध्ये तुम्ही बघू शकता. या दोन टेम्पलेट स्ट्रँड पैकी हा जो येलो कलरने दाखवलेला टेम्पलेट आहे त्याचं 3' end हे फ्री आहे. And this template strand which has free 3' end is called as leading template. It is called as leading template. And the new strand synthesized on leading template is known as leading strand. आणि या leading template वर तयार होणाऱ्या नवीन स्ट्रँडला leading strand असं म्हणतात. Why? Because this new strand is synthesized quickly, earlier, continuously and towards the replication fork. कारण हा जो न्यू स्ट्रँड आहे लीडिंग टेम्पलेट वर तयार होणारा तो कंटिन्युअस तयार होत जातो, आधी तयार होतो आणि तो रेप्लिकेशन फॉर्क कडे तयार होत जातो. म्हणजे द सिंथेसिस ऑफ न्यू स्ट्रँड ऑलवेज टेक्स प्लेस फ्रॉम 5' to 3' end, while the other template strand is known as lagging template. Lagging template means it has free 5' end and the new strand synthesized on A, याच्यावर तयार होणारा जो न्यू स्ट्रँड आहे that new strand is known as lagging strand. Please don't confuse. The template strand which has 5' end is known as lagging template and the new strand which is synthesized on lagging template is known as the lagging strand. Why it is called lagging strand? Because this lagging strand is synthesized slowly, late and discontinuously, that is in the form of short segment of nucleotides and away from the replication fork. अर्थात हा जो स्ट्रँड आहे लॅगिंग स्ट्रँड हा हळूहळू नंतर तयार होतो आणि तो रेप्लिकेशन फॉर्क पासून दूर जात असतो, म्हणून त्याला लॅगिंग स्ट्रँड असं म्हणतात. नाउ हियर फॉर द सिंथेसिस ऑफ दिस each segment of lagging strand always it requires new RNA primer, and these fragments of lagging strands are called Okazaki fragments. या fragments ला Okazaki fragments असं म्हणतात. Of course, this name is given after scientist Okazaki. Later on, these RNA primer are removed by the action of enzyme DNA polymerase first, and they are replaced by nucleotide sequence so that Okazaki fragments get interconnected to each other in presence of enzyme DNA polymerase first in prokaryotes and DNA polymerase alpha in eukaryotes. In this manner, on both of these old strands, the new strands are synthesized, and at the end of replication, two daughter DNA molecules are formed with one old strand and one new strand. Of course, the coiling of both strands is again carried out by means of enzyme topoisomerase or gyrase. So here we have completed the study of DNA replication. Thank you.